All right, deep divers, get ready. Because today we are going to explore preclinical efficacy models. We're going to unpack how scientists determine if a new drug actually works, before it even gets to a point of human trials.
Yeah.
And you've sent us some really fascinating stuff this time, journal articles, FDA docs, even some case studies from your own work at OPR&D.
Yeah, it really is like piecing together a puzzle. We gather evidence from different sources to build a case either for or against a drug's potential.
I love that analogy. And today's puzzle pieces come from two main sources, in vivo and in vitro models.
Exactly. In vivo refers to testing on living organisms, and often this means animal models, mice, rats, even primates sometimes. And in vitro involves controlled environments, so things like test tubes or cell cultures.
It's kind of like testing a new fertilizer on a single plant in a pot before you try it on a whole field.
Precisely. Each approach offers unique insights, but just like you said with your analogy, they also have limitations.
That makes sense. So let's just start with the basics. Why are these preclinical efficacy models so crucial? And we can't just skip ahead to human trials, right?
Absolutely not. Imagine building a skyscraper without testing the foundation first. That's essentially what it would be like to bypass preclinical testing, so we need to gather enough evidence to justify the risk and the expense of moving to human testing.
Okay, that's a great way to put it.
Yeah.
So then how do scientists choose between in vivo and in vitro? Is it like choosing the right tool for the job?
You hit the nail on the head. The choice depends on a lot of different factors. The disease that's being targeted, the drug's mechanism of action, basically how it's supposed to work at the molecular level, and of course, ethical considerations, especially when we're talking about using animals.
Speaking of which, those OPR&D cases that you sent in, I noticed a few of them involved preclinical models for Alzheimer's disease, and those always seem particularly complex.
They are complex. One case that you sent in detailed a study using a mouse model that was genetically engineered to develop amyloid plaques. And those are those protein clumps that are found in the brains of Alzheimer's patients.
Ah, so the researchers created a model that closely mimics a key aspect of the human disease.
Exactly, and then they tested a potential drug's ability to reduce those plaques, and importantly, they observed the effects on the mice's cognitive function.
It's so fascinating how scientists can tailor these models to specific diseases.
It is, but we always have to remember no model is perfect, and that's where this idea of predictive power comes in.
Okay, tell me more about that. I'm guessing it refers to how accurately a model predicts what will happen in humans.
Exactly. A model with high predictive power gives us more confidence that a drug's effects that are observed in the model will translate to humans.
But I imagine that can be tricky to assess, right?
It can be tricky. Take those Alzheimer's models, for instance. Mice and humans have very different brains, both structurally and functionally. So while a drug might reduce plaques in a mouse, it doesn't guarantee it will do the same thing in a human, let alone lead to improvements in cognitive symptoms.
Right. That highlights a key point you made earlier. Every model has its limitations. What are some of the other challenges that researchers face?
Well, with in vivo models like using animals, there are ethical considerations. Of course, we have to keep those in mind always, and then they can be really costly and time-consuming as well.
That makes sense. What about limitations of in vitro models?
One major drawback is oversimplification. Testing a drug on cells in a dish can't fully replicate those intricate interactions that are happening within a living organism.
It's like trying to understand a symphony by listening to just a single instrument.
A perfect analogy, you might miss the nuances and the complexity of the full composition.
So a drug might work great in a test tube, but then fail in an animal because it can't actually effectively reach its target within the body.
Exactly. These limitations really underscore why researchers carefully consider both the strengths and the weaknesses of each model they're using, often using multiple models to get a more comprehensive understanding.
That brings me to another point. I noticed several FDA and ICH guidelines in your materials. I'm guessing those play a big role in making sure that these models are being used effectively.
Absolutely. Regulatory bodies like the FDA and the International Council for Harmonization, they set really strict standards for preclinical studies, and this ensures data quality, ethical conduct, and ultimately patient safety.
So they're setting the bar high for what constitutes, reliable and meaningful results?
Precisely. For instance, there are very specific guidelines on selecting and validating animal models, designing experiments, and even how they're collecting and analyzing the data, and all these measures are aimed at reducing bias and increasing the reliability of preclinical research.
That makes sense. So it's not just about doing the research, it's about doing it rigorously and transparently. Those OPRND cases you sent in, do any of them offer a glimpse into how these guidelines play out in real-world research?
They do. One case involved a new antibiotic, and the researchers initially tested the drug's bacteria-killing ability in a petri dish, which is a standard in vitro approach.
Makes sense, but I'm guessing they didn't stop there.
You're right. They also tested the drug in mice that were infected with the same bacteria to observe its effects within a living system. This multifaceted approach, guided by those regulatory standards, helps researchers build a much stronger case for a drug's efficacy.
It's like using multiple lenses to examine a specimen to gain a more complete picture.
Exactly. By combining in vitro and in vivo data, researchers can identify potential benefits and risks early on, and this allows them to make better, more informed decisions about which drugs should move forward.
So it's a crucial step in that whole drug development journey. And understanding these models is essential for anyone who's following the latest medical advancements.
Absolutely.
Now let's dig a little deeper into this concept of efficacy. How do scientists actually measure if a drug is working in a preclinical setting?
That's where things get really interesting. Well, it all comes down to, really understanding what efficacy actually means, in the context of preclinical research. So we're not just looking for any effect. We're searching for effects that are likely to be meaningful in a clinical setting. So, effects that will translate to real benefits for human patients.
So it's not just enough to see a drug working in a petri dish or even in a mouse.
Uh,
You need to be confident that it will have a real impact on human health.
Precisely. And that's where that concept of predictive power that we talked about earlier really comes in. We want to know how well those preclinical results, so from both the in vivo and the in vitro studies, predict what will actually happen when the drug is then tested in humans.
And I'm guessing that's not always easy to determine. How do scientists actually go about measuring efficacy in these preclinical models?
Well, it really depends on the disease and the specific drug that's being investigated. Sometimes it's pretty straightforward, like in a cancer model, for instance. We might measure tumor size reduction as, a clear indicator of the drug's effectiveness. Or in an infection model, we could measure the reduction in bacterial counts.
Okay, those seem like pretty clear-cut endpoints, things that are easy to measure and directly relevant to the disease.
They are. But in other cases, measuring efficacy can be much more complex. Think back to those Alzheimer's models that we were talking about earlier. Measuring a drug's impact on cognitive function is far more challenging than simply measuring tumor size.
That makes sense. Cognition is such a multifaceted aspect of brain function.
Exactly. It's not always easy to pinpoint precisely how a drug is affecting those intricate cognitive processes, even in a controlled preclinical setting.
So how do researchers tackle that challenge? Are there specific methods for measuring these more nuanced effects?
Researchers often develop very specialized behavioral tests that assess various aspects of cognition, so things like memory, learning, attention. And they very carefully observe the animal's performance on these tasks to gauge whether the drug is having a positive impact.
It sounds like they're almost becoming animal psychologists, carefully studying behavior to understand the drug's effects.
Yeah, that's a great way to put it. And sometimes they even use brain imaging techniques, similar to those used in human studies, to observe changes in brain activity in response to the drug.
Wow, that's incredible.
Yeah.
It really highlights the ingenuity and the dedication that's required to assess efficacy in these complex disease models. But even with these very sophisticated techniques, there's still a level of uncertainty, right? We can't be 100% certain that what we see in a preclinical model will perfectly mirror what happens in humans.
You're absolutely right, and that's where things can get even more complicated. Sometimes researchers have to rely on what are called surrogate endpoints, and these are measurements that are thought to correlate with the actual clinical outcome that we're interested in, but they're not the outcome itself.
Okay, that sounds a bit abstract. Can you give me a more concrete example of a surrogate endpoint?
Sure, imagine a study of a new drug for heart disease instead of directly measuring the drug's ability to prevent heart attacks, which, would be a very long and complex study. Researchers might use cholesterol levels as a surrogate endpoint because we know that high cholesterol is a risk factor for heart attacks. So if the drug effectively lowers cholesterol, it's reasonable to assume that it might also reduce the risk of heart attacks down the line.
It's like using a proxy measure, something that's easier to measure in the short term, but that's believed to be a good indicator of the long-term outcome that we're ultimately interested in.
Exactly. And surrogate endpoints can be incredibly valuable tools in preclinical research. They allow scientists to get a much quicker read on a drug's potential without having to wait for those long-term clinical outcomes to unfold.
But I can also see how relying on surrogate endpoints could be a bit tricky. What if the correlation isn't as strong as we thought? What if a drug improves the surrogate endpoint, but it doesn't actually translate to a meaningful benefit for patients?
You've hit on a very critical point, and that's why the selection and validation of surrogate endpoints is so important. Researchers need to be really careful in choosing endpoints that are truly reflective of that clinical outcome they're interested in, and they need to back up those choices with solid scientific evidence.
It sounds like a delicate balancing act, using surrogate endpoints strategically to accelerate research, but also being very aware of their limitations and potential pitfalls.
Precisely, and that's where those FDA and ICH guidelines that we discussed earlier come back into play. Those guidelines provide valuable guidance on how to choose and validate surrogate endpoints appropriately, ensuring that preclinical research is being conducted rigorously and ethically.
So those guidelines are almost like a safety net, helping to make sure that the research is on the right track and that the conclusions drawn from it are sound.
Exactly. They really help to minimize bias, increase transparency, and ultimately protect patients by ensuring that only the most promising and well-supported drugs actually move forward in the development process.
Okay, that makes a lot of sense. Now, I know you've been diving deep into those OPR&D cases from our listener. Have you come across any examples that really illustrate these complexities of measuring efficacy and, navigating the world of surrogate endpoints?
Yeah, I have one case that really stood out, involved a company developing a new drug for Parkinson's disease, and they were using a rat model that mimicked some of the key motor symptoms of the disease, so things like tremors and rigidity.
So they were working with a model that closely reflected the clinical presentation of the disease in humans. What were they looking at as their measure of efficacy?
They were actually taking a multi-pronged approach, which is often, the most informative. They were observing the rat's behavior, looking for improvements in those motor symptoms, but they were also looking deeper, at the cellular level, specifically at the dopamine-producing neurons in the brain.
Because those dopamine neurons are progressively lost in Parkinson's disease,
Right.
leading to those very debilitating motor symptoms.
Exactly. So the researchers were measuring both behavioral changes, the outward signs of improvement, but also cellular changes, those underlying biological mechanisms that are at play.
That sounds like a very comprehensive way to assess efficacy. What did they find?
Well, they actually observed some pretty encouraging results. The drug significantly improved the rat's motor function and then also showed a protective effect on those crucial dopamine neurons.
Wow, that's really promising. It sounds like a potential breakthrough for Parkinson's treatment.
It certainly did, but what's really interesting about this case is how the researchers approached the data. They were very cautious in their interpretation, acknowledging that while the results were very exciting, they were still just one piece of the puzzle.
So they weren't getting ahead of themselves, recognizing that success in a rat model doesn't automatically translate to success in humans.
Exactly. They emphasize the importance of further research, including clinical trials in human patients, to confirm these initial findings and to fully understand the drug's potential benefits and risks.
That's a great example of the scientific process in action, celebrating the successes but also maintaining a critical eye and recognizing the limitations of each step along the way.
Precisely. And it highlights the importance of transparency in research, being upfront about both the strengths and weaknesses of a study and not overstating the findings. This honesty and transparency are really essential for maintaining public trust in the scientific process and ultimately ensuring that only the most promising and well-supported drugs advance to human trials.
Okay, so we've talked about measuring efficacy in preclinical models, but let's zoom out a bit. We've mentioned these FDA and ICH guidelines several times. Can you get a little more insight into the specific role these guidelines play in shaping preclinical research? What are some of the key areas that they address?
They do, these guidelines, they cover a really wide range of aspects, but they're all aimed at ensuring the quality, reliability, and ethical conduct of those preclinical studies.
So they're not just focused on those scientific aspects, they're also addressing those ethical dimensions of, working with animal models.
Absolutely. Ethics is a paramount concern in preclinical research. And, these guidelines provide very clear guidance on how to minimize the number of animals that are used, how to ensure their welfare, and how to use, appropriate anesthesia and analgesia to minimize any pain or distress that they might experience.
That's reassuring to hear. It's good to know that those ethical considerations are really kind of woven into the fabric of that research process.
Yeah, they are. And beyond the ethical aspects, the guidelines also address a lot of the nuts and bolts of designing and conducting, high quality studies.
Okay, tell me more about that. What are some of the specific areas that they delve into?
Well, they provide really detailed guidance on things like selecting the right animal species and strain for a particular study, making sure that the animals are housed and cared for, in a way that really meets their specific needs, and even controlling for variables that could influence the results of the experiment.
So it's kind of like creating this standardized playbook for preclinical research, making sure that everyone is playing by the same rules and that the results are, as reliable and as meaningful as possible.
Exactly. The guidelines also delve into, statistical considerations, outlining appropriate methods for analyzing data, and ensuring that the conclusions that are drawn from the research are statistically sound.
So it's not just about collecting data, it's about analyzing it rigorously and making sure that the conclusions are really well supported by that evidence.
Precisely. And they even address data integrity, ensuring that all data is accurately recorded, stored, and reported.
Wow. It sounds like these guidelines really cover every aspect of preclinical research, leaving no stone unturned in the quest for, quality and reliability.
Yeah, they really do. They provide a framework for conducting research that's both scientifically rigorous and ethically sound, and ultimately, that benefits everyone, the researchers, the regulators, and most importantly, the patients, who will one day benefit from the new drugs and treatments that emerge from this research.
Absolutely. It feels like we've journeyed to the heart of drug discovery, examining those crucial early stages where scientists are gathering evidence and really building a case for a drug's potential.
Yeah, it's like we've been working right alongside those researchers, piecing together that puzzle of preclinical efficacy.
Exactly. And speaking of puzzles, I have one for you, deep diver. As you continue exploring those research papers and those reports, you're going to encounter a sea of data graphs, tables, statistics. It can feel really overwhelming at times. So what's the key to navigating all that data and actually extracting meaningful insights?
That's a great question. And it really all comes down to, developing a critical eye for data. So don't just passively absorb those numbers, question them, interrogate them, figure out what they truly mean and what story they're really telling.
So it's not just about, seeing a graph that shows a drug reducing tumor size. It's about understanding how that data was collected, what the units of measurement are, and whether those findings are statistically significant and reliable.
Precisely. And remember, data can be presented in so many different ways, and some of those ways are much more transparent and informative than others. So pay close attention to those graphs and those tables. Are they clearly labeled? Do they accurately represent the data, or are they potentially misleading, designed to highlight certain findings while kind of obscuring others?
It's like being a detective, carefully examining the evidence for any signs of manipulation or distortion.
Exactly. And as you kind of delve deeper into those OPR&D cases that you sent, you'll really see how data interpretation plays a really crucial role in, real-world research. Sometimes the story that the data tells isn't as straightforward as it might seem at first glance.
Oh, I can imagine. I bet those case studies offer some pretty fascinating examples of, how data can be used and sometimes even misused in the world of drug development.
Oh, they do. One case that really comes to mind involved a company that was developing a new obesity drug, and their preclinical studies in mice yielded some really impressive results, significant weight loss, improved metabolic markers. It looked like a potential blockbuster.
So it sounds like they had stumbled upon a miracle cure. What happened?
Well, when the drug moved into human trials, the results were much less impressive. The weight loss was really minimal, and there were actually some concerning side effects. And it turned out that the company had basically cherry-picked their preclinical data, highlighting only the positive findings while downplaying or even ignoring the negative ones.
Wow, that's not only misleading, it's potentially really dangerous. Patients could be given false hope and resources could be wasted pursuing a drug that's ultimately ineffective or even harmful.
Yeah, you're absolutely right. And this case really underscores the importance of transparency and rigor in preclinical research. It's crucial to report all the data, both the positive and the negative, and to be upfront about the limitations of the studies. This honesty and this transparency are absolutely essential for maintaining public trust in the scientific process and ensuring that only the most promising and well-supported drugs actually advance to those human trials.
It's a good reminder that even in the world of science, where objectivity is paramount, there can be those subjective biases and agendas at play.
Hmm.
We need to be aware of those potential biases and approach data with a healthy dose of skepticism.
Absolutely. And that's where, critical thinking really comes in as you read those research papers. Don't just accept those conclusions at face value. Dig deeper, ask questions, consider the source of the information and any potential conflicts of interest that might be there.
It's about being an active participant in that process of knowledge acquisition.
Hmm.
You're not just a passive recipient of information.
Precisely, and remember knowledge is power. By really understanding how preclinical efficacy models work, recognizing both their strengths and their limitations, and learning to interpret data critically, you're empowering yourself to make better, more informed decisions about your health and your well-being.
And that's what this whole deep dive is all about, empowering you, a deep diver, to navigate this really complex world of science and medicine with confidence and a discerning eye.
It's about giving you the tools you need to understand not just what the research says, but also how that research was conducted, what it truly means, and how it might impact your life.
We've covered a lot of ground in this deep dive, from those fundamentals of preclinical models to the nuances of data interpretation and the crucial role of regulatory oversight. But as with any deep dive, there's always more to explore.
There is. And as you continue your exploration, just remember that the world of science is constantly evolving. New discoveries are being made, new technologies are emerging, and our understanding of the human body and disease is constantly deepening.
So keep asking questions, keep challenging those assumptions, and keep that critical thinking cap firmly in place.
And never lose sight of the ultimate goal of all this research: to improve human health, to alleviate suffering, and to extend the reach of human potential.
On that note, we'll leave you with a final thought. We've talked a lot about predicting a drug's effectiveness in humans, but what about predicting its impact on the broader environment as we develop these new drugs?
Yeah.
We need to consider not only their effects on human health, but also their potential impact on those ecosystems we all share. That's a challenge for science and for society as a whole, and it's something worth pondering as we strive to create a healthier and more sustainable future for everyone.
That's a really profound thought and a fitting reminder that our pursuit of scientific knowledge should always be guided by a sense of responsibility and a commitment to the well-being of both humanity and the planet we all call home.